Whenever bacteria or pathogen comes to our mind, the first name which strikes is Escherichia coli or Staphylococcus aureus or Streptococcus pneumoniae. But today in this presentation, we will discuss about a deadly pathogen which causes a number of nosocomial infections and health-related diseases, that is Streptococcus pyogenes. Now, moving on to what is this bacteria Streptococcus pyogenes. Streptococcus pyogenes is a bacterial pathogen, as I mentioned previously, which causes a number of diseases ranging from mild localized infections, that is, which are not deadly, to life-threatening diseases and nosocomial infections. Now, Streptococcus pyogenes is a gram-positive, aerotolerant bacteria. What are they made of? They are made up of non-motile and non-spore forming cocci. What does this cocci signify? This cocci signify round-shaped cells. In Streptococcus pyogenes, these round-shaped cells or cocci are arranged in chains. Now, from where is this name Streptococcus pyogenes is derived? It is derived from a Greek word where streptos means a chain and coccus means berries or coccus. Now, it is also related with pus. Why we are saying this bacteria is related to pus? Because it causes a number of infections which produce pus-like or fluid-like substances. Streptococcus pyogenes is also known as GABHS. What does that signify? It signifies Group A beta hemolytic streptococcus. Now, what is this Group A beta hemolytic streptococcus? It shows beta hemolysis. What is beta hemolysis? Beta hemolysis is a phenomenon where clear zones are formed around the colonies in blood agar medium. Why do they show these clear zones? Because this pathogen shows complete hemolysis and is able to digest or break down the blood cells which are present in the blood agar medium. Moreover, the classification will be further clear in the next slides. Now, who is this person, Theodore Billroth, or what has this scientist contributed to? We will see in the upcoming slide. Now, when did we come to know about the streptococcal infections or the various diseases which are caused by Streptococcus pyogenes? As we have seen the picture of Theodor Billroth, he is an Austrian surgeon who in the year of 1874 attributed or discovered the various causes of streptococcal infections. How did he describe these organisms? He described them as small organisms which are found in either isolated form or they are arranged in pairs, sometimes in chains of 4 to 20 or more links. Now, when did we come to know about the real importance or the virulence or the deadly cause of the microorganism? It was depicted by the scientist Louis Pasteur. How did he depict? He isolated the microorganism from the uterus and blood of women who were suffering from puerperal fever. Now, this fever has a deadly infection and may lead to various deaths of various people. Thus, it indicates it can cause a number of deadly diseases. Hence, it also depicted that the microorganism Streptococcus pyogenes can cause a number of infections and a number of deadly diseases. In this particular slide, the taxonomical order of the organism Streptococcus pyogenes is provided where the domain, kingdom, phylum, class, ordo, family, genus is depicted. Classification. Classification of the organism Streptococcus pyogenes can be done on two bases. Basis of hemolysis and Landsfeld system. Now, on the basis of hemolysis, how can they be classified? They can be classified into alpha, beta, gamma. In alpha hemolysis, a green zone of discoloration around colonies is observed. Now, which organism shows this kind of hemolysis? Viridans streptococci. Beta hemolysis. In beta hemolysis, a clear zone of hemolysis is observed. which is a characteristic property of obviously Streptococcus pyogenes and also Streptococcus hemolyticus. In gamma hemolysis, there is no color change of the medium or there is no sign of hemolysis. It is shown by Streptococcus faecalis. Now, how did Lancefield classify? Lancefield was able to demonstrate over 50 different M types. Now, what are these M types? M type is a protein present on the surface of the streptococcal pyogenes of group A streptococci during her career. After that, 200 M types were discovered on the basis of various serological studies and experiments. Moving on, a picture is given on how we observe the various hemolysis, alpha hemolysis, beta hemolysis and gamma hemolysis. As you can see in alpha hemolysis, greenish discoloration is observed with partial hemolysis. Beta hemolysis, we can observe clear zones, hence complete hemolysis. And in gamma hemolysis, there is no sign of hemolysis, no colonies are formed. Epidemiology. Now, what does this word epidemiology signify? Epidemiology signifies that the microorganism Streptococcus pyogenes is able to colonize in what parts of human body and cause a particular infection or disease. So, what are the areas which are affected by Streptococcus pyogenes? Streptococcus pyogenes mainly colonizes or causes infection in the throat, genital tract and skin. Now, how are these bacteria transmitted? They can be transmitted in various ways, such as inhalation of respiratory droplets, skin contact, droplet particles, through food, dust, and various toxins. It can cause a number of diseases. What are the basic diseases which are caused by Streptococcus pyogenes? They include streptococcal pharyngitis, scarlet fever, and heart diseases. Now, who are the people or what are the areas which are at various risk? For example, in healthy individuals, 1% to 5% have throat, vaginal or rectal carriage. In healthy children, such carriage rate varies from 2 to 17. Moving on to the morphology of the organism Streptococcus pyogenes. As I previously mentioned, in Streptococcus pyogenes, the cocci are linked in the form of chains. Now, the chains can be 5 cocci or more. These type of bacteria produce capsules. In group A strains, capsule is hyaluronic acid in nature. They consist of M proteins, as I previously mentioned in Lancefield classification, which are of major importance because it is antiphagocytic. Now, what does this word antiphagocytic mean? It means that M proteins prevent phagocytosis of the bacteria. It means it prevents the immune system to digest the bacteria and hence they can cause the disease and it is directly involved in virulence. How does the growth of this bacteria occur? The growth occurs by elongation on the axis of the bacteria parallel to the chain and the division takes place in right angles at the equatorial plane. They can also divide asexually by binary fissions or they can divide sexually by forming gametes. Here in this picture, a gram stain picture of Streptococcus pyogenes is provided on the left hand side and on the right hand side a hemolysis picture is shown of the organism where Streptococcus pyogenes shows beta hemolysis or clear hemolysis of the red blood cells. Moving on to the antigenic structures. The antigenic structures are the structures which are present on the surface of pathogenic bacteria or which are responsible for causing or adherence to the host cell. First is the M proteins. The M proteins are chief virulence factor of the group A streptococci. As I previously mentioned, they are antiphagocytic, which means they can resist phagocytosis by polymorphonuclear leukocytes. Now, what are these polymorphonuclear leukocytes? They are leukocytes having more than one nucleus. As the phagocytosis is prevented, hence the immune system is weakened and the bacteria can multiply rapidly in human blood and cause the infection. Moving on to T proteins. T proteins do not have any relationship with the virulence of streptococci. R proteins, R antigens can cause cross reactions in grouping or typing antisera that contain antibodies to one or other of them. Lipoteichoic acid. Now, what is the role of lipoteichoic acid? Lipoteichoic acid plays a role in host bacterial infection in which it helps in assisting the bacteria to the host cell. Nucleoproteins. Nucleoproteins are also called P substances. Like T proteins, they do not have any serological specificity or any relationship with the virulence of streptococci. Next is hemolysin. Now the pathogen consists of two types of hemolysins, oxygen labile and oxygen stable. Now they are streptolysin O and streptolysin S. Streptolysin O is responsible for hemolysis in the deeper layers of blood agar where streptococci grow in the absence of oxygen. I repeat, in the absence of oxygen. Streptolysin S is responsible for hemolysis on the surface layers of blood agar where streptococci go in the presence of oxygen. Next is the pyrogenic toxin. Now, streptococcal pyrogenic substances or toxin weaken the immune system, hence the susceptibility towards endotoxic shock and alterations in the T cell function increases. Now, as the T cell function is alterated, hence the immune system is dampened or damaged. Hence, the organism can cause the disease at an increased incidence. Last but not the least is hyaluronidase, which is liberated by group A streptococci, which splits the organism into two and helps them in spreading and multiplying in the bacteria in the human blood, and hence they can cause the disease at a greater incidence. This slide represents the process of pathogenesis or pathogenicity of the organism. These few steps are common in all the microorganisms for causing a particular infection or a particular disease in the host cell. First is adherence. In adherence, the bacteria attaches itself to the mucosal surfaces of the host surface or the cell wall of the host surface. In invasion, the bacteria invades the epithelial cells or deeper cells of the human body. Evasion is the process of evasion of the immune system via C5a peptidase. Now, this C5a peptidase is an enzyme which is present in the bacteria Streptococcus pyogenes, which helps it to evade the immune system, which consists of opsonization and phagocytosis. Finally, the bacteria infects the human with its pathogenic factors, production of various toxin and enzymes. Here in this chart, a pathogenicity of the Streptococcus pyogenes is provided. In the first case, the Streptococcus pyogenes produces a super antigen toxin which further releases cytokine and further causes toxic shock syndrome. Toxic shock syndrome may also be caused by pyrogenic toxin. In enzyme release, it leads to local inflammation of invasion tissues, which further leads to skin infection, pharyngitis. It may also cause bacteremia, which indicates multiplication of bacteria in blood, leading to meningitis and osteomyelitis. Streptococcus pyogenes may also cause post-streptococcal glomerulonephritis and acute rheumatic fever. This slide shows a list of people or person who are at risk groups for the diseases which are caused by Streptococcus pyogenes. Now, these people are not only at risk factors for the disease which are caused by Streptococcus pyogenes, but also other bacteria. They include immunocompromised host, people undergoing major surgeries, organ transplantation in the recent time, infants, people suffering from primary infection, various allergies like food, pollen and other pollens. Moving on to the diseases which are caused by Streptococcus pyogenes. Firstly, local infections. They include sore throat and pyoderma. Now, what is pyoderma? Pyoderma is the occurrence of red lesions and ulcers on sore throat, on legs, hand, and back of the neck. Invasive diseases include sepsis and toxic shock syndrome. Both of these diseases have deadly infection on human. Sepsis is the multiplication of toxins in blood, and toxic shock syndrome is caused by pyrogenic toxin. Infective endocarditis, as the name suggests, endocarditis is the inflammation of the heart. It may also include meningitis. Now, what are the post-streptococcal diseases? The post-streptococcal diseases may be asymptomatic and the person may be a carrier. It includes acute rheumatic fever and acute post-streptococcal glomerulonephritis. Now, in rheumatic fever, it is indicated streptococcal pharyngitis, that is the inflammation of the pharynx. The mortality rate increases when heart disease takes place. In streptococcal glomerulonephritis, edema, seizures, heart failure takes. Here, a list of various Streptococcus pyogenes group A diseases are listed, which are also known as GAS. Some of the diseases include superficial diseases like pharyngitis, vaginitis, deep infections like bacteremia, meningitis, toxin-mediated, as we already saw, toxic shock-like syndrome, immunologically mediated reactive arthritis. On the left-hand side, a figure is provided which indicates the various areas which are affected by the group A Streptococcus pyogenes. Moving on to the laboratory diagnosis of the organism. Now, how is this organism indicated that it causes a particular disease? The 3 or 4 techniques include direct demonstration. In direct demonstration, smears are prepared of the organism. Now, how are these smears prepared? They include the specimens such as pus, wound exudate, showing single cocci or pairs rather than definite chains. In culture and identification, as we have already seen, that the characteristic feature of group A streptococci is that they show beta hemolysis. Now, beta hemolysis is the formation of clear zones around the colonies. As a result, the organism can be depicted. Antigen detection tests can also be run, like latex agglutination and coagglutination kit. Last but not the least, serological tests can also be done for bacterial diagnosis. In these tests, they detect the antibody level to extracellular products which are liberated by the bacteria Streptococcus pyogenes. If the antibody level is normal, then no disease is depicted. But if the antibody level is higher than the normal antibody level, then there may be a disease factor or disease risk. Moving on to the treatment of the particular organism or the disease which is caused by Streptococcus pyogenes. All groups of streptococci are sensitive to penicillin G and most of them are sensitive to erythromycin. Now, people who are allergic or have some problems related to health issues or who are sensitive to penicillin G are instructed erythromycin. Clindamycin and vancomycin may also be used. Below the chart shows the mechanism of the various antibiotics by which they kill the microorganism. Penicillin, as we all know, mechanism is interferes with the synthesis of a peptide in the bacterial cell wall. Clindamycin stops or inhibits the RNA-dependent protein synthesis. Vancomycin also works by the same mechanism. Vaccines may be used, but penicillin G and erythromycin is the smart choice. Moving on to prevention. Now, some of the prevention are the basic rules of our life, like personal hygiene, cleanliness, equipment sterilization, sharing of personal stuff should be avoided, cleaning of various hospital equipment like catheters, knives, scissors, etc. Surgeries or drugs that compromise the immune system may be avoided by taking medicines. Exposure to smoking, drinking, or various pollen grains or allergies must be avoided. Last but not the least, some recent discoveries on the organism Streptococcus pyogenes is provided. Streptococcus pyogenes Cas9 protein is the most common type of protein used in the type 2 CRISPR-Cas9 system. Now, what is this CRISPR-Cas9 system used for? They are used for gene editing tool which modifies or rectifies the genome of a particular organism so that they can be used as research models or various discoveries. Homologs of SpiceApp have been discovered. Now, the SpiceApp is a serine protein domains or multi-domains which have virulence factors. The SpiceApp helps in the pathogenicity of the organism, thus they can cause various diseases. Some other facts on recent discoveries are provided, and last, a reference slide is provided for the PPT presentation.